Good morning, everyone. My name is Francesco Pignatti. I'm head of the Office of Oncology Evaluation at EMA. I'm very, very pleased to welcome you to this workshop. Paolo, we started the idea about this workshop about a few months ago, 4 or 5 months ago, and we told you that perhaps there is an opportunity to advance in this difficult field, and you jumped on it, and it was for us a tremendous motivation for moving forward. So I'd really like to thank you for enabling this collaboration, and perhaps you will tell us what you see in this workshop and how we're going to spend the next seven hours. What do you think we can get out of it?
Thank you, Francesco. Indeed, it's a privilege for us to be here, and I welcome all of you on behalf of ESMO, the European Society for Medical Oncology, on behalf of its president, Fortunato Ciardiello. The past president of Style is here. The president-elect, Josep Tabernero, has been deeply involved in the preparation of this meeting. Today, there is a meeting in Barcelona on GI cancers, and so he cannot attend. Yes, personally, I'm a medical oncologist following one kind of rare cancers, which is sarcomas. And so this is the only reason why I've been involved in the past in the problems which have to do with rare cancers, also in a way from the political point of view. And you know that ESMO coordinates Rare Cancers Europe, which is a multi-stakeholder effort with the scientific societies, with the patient groups, and with the industry inside. So we try to be multidisciplinary in order to address the many problems of rare cancers. Clearly, rare cancers are a model, I think, for some of the issues which are ahead of us now in medical oncology, but from the beginning we agreed that this meeting was half about rare cancers, half about personalized oncology, and Josep Tabernero was deeply involved in the preparation also for this reason, because of course when the numbers become small, even in frequent cancers, the problem are more or less the same. But the main issue is, I think, that, of course, today we are talking about regulatory issues. What would be important for us is that these regulatory issues could be seen also in a clinical perspective, because in the end they, of course, affect our profession, our everyday professional life. And maybe we as oncologists have been too far from these issues, from the methodological issues about research and about the clinic, which are implied. And ESMO has tried to do something in the last years, in particular the magnitude of clinical benefit. We tried to develop a scale to raise the bar, so to join the idea of raising the bar, of course, but also trying to do this from a clinical perspective. And I'm happy that Elisabeth De Vries is here, who is following this item for ESMO, within the ESMO committee devoted to this topic. And so I think it's a sign that the oncology community, the community of clinicians, is more involved now in addressing these methodological issues, which then result into regulatory issues, and then regulatory guidelines, and then in the regulation of new drugs in Europe. So I'm very happy to be here, of course. I thank deeply EMA, because we talked in these years several times about having this kind of meetings together among clinicians and regulators, just to to close the triangle of regulators, the industry, and clinicians and patients. And here also patients are represented in this room. So I very much hope that this meeting can be useful, actually useful. Thanks, Francesco.
You're very welcome. I've put together a few slides just to try to frame the problem. And let me start by saying that it's not really about problems that I think we should be spending the next 7 hours discussing, but it's more about the solutions. 1 year ago, when the germ of this workshop started, I was sitting with my colleague Jorge Martinalbo at a scientific advice, and we were really puzzled by the range of views that there were about feasibility of randomized studies. Are they even necessary in a particular situation when the phase 2 data are so convincing, et cetera, et cetera. And we were wondering, is there a way that we could further the debate and maybe consolidate on some common principles that we could perhaps elaborate into guidance and so on? And things, even in 1 year, have progressed. I think we have become more confident with a number of approaches. But we can surely all agree that randomized active and placebo-controlled trials, whenever feasible and whenever they make sense, are the most convincing evidence generation mechanism that we have. We also know that for most orphan drugs approved by EMA, randomized controlled trials were actually possible to conduct. So these are things that I think we can all agree. But what about the situation where these are not feasible? And we distinguish mainly about two situations, one where the population is too small, and the other one when the evidence is too compelling. How do we deal with these situations today? There's a little bit of guidance, but really we deal with them rather retrospectively, looking at the evidence, how good is the evidence, and then taking some sort of decision. Is there a possibility to become more systematic about these type of situations and the approach? Now, one of the two, the so-called dramatic effect, is a relatively easy one. And we think we can all agree that even in the absence of randomized controlled trials on the use of parachutes, that there is little uncertainty that would warrant more evidence to be generated on the use of parachutes instead of no parachutes. But is it always so clear? Unfortunately, in many in drug development, the situations are much less clear-cut. So what is dramatic? Do we have any type of grasp or principles that we could agree on defining this concept? And then based on what parameters, because that's always a tricky issue, and to make these issues a little bit more explicit and explainable. Okay, what is self-evident? It's difficult to use this concept in practice and be informative. There is, as I said, a little bit of guidance on how to deal about situations where externally controlled trials are necessary or considered useful. But these are relatively old guidance. Perhaps we have moved forward with the type of evidence that can be used to generate these external controls. And then can we be a little bit more specific? Perhaps oncology has its own peculiarities. And lastly, there is this tension. While we think we are driven by the concept of making sure that drugs are made available as quickly as possible, there is this tension between this aim and the aim of having as complete information as possible. So we need to make sure that all those different stakeholders have slightly different objectives, but that the whole system works together. And it doesn't make sense to approve drugs so early that then clinicians and patients cannot make decisions about benefits or risks, or health technology assessments cannot do their work. So we need to be thinking a little bit together with other stakeholders. And lastly, let's remember the primary aim of a clinical trial must not be evidence generation per se. It must be that it's treatment, the optimal treatment for a patient. So this is something that we always need to take into account. And we see, for sure, many of the methods and the methodological guidance would dictate a certain design, but then perhaps this is totally undesirable for patients. And this must be the driver for what we consider acceptable design. So clearly, lots of people need to be at the table to discuss this type of issues. Finally, why are we having this workshop today? I think if you look across the recent experience, a number of significant drugs have been approved on the basis of single-arm trials, so we have a lot of experience, and there is much more happening in terms of data sharing, libraries of data and so on, that we can use to complement clinical data from trial. So perhaps there is an opportunity to consolidate this, and in 7 hours' time, when Dr. Casali, you will wrap up, I hope that we will have some concrete examples or where we could move forward and consolidate some agreement on the main principles.
Okay, thanks, Francesco. Now we can start with the first session, which has to do with scenarios and case studies. So the first speaker will be Mace Rothenberg, Chief Development Officer, Oncology Pfizer Global Product Development, and he will talk about Crizotinib in patients with ROS-positive non-small cell lung cancer. Please.
Thank you, Professor Casali and Professor Pignatti, and to the other organizers. Thanks so much for pulling all of us together for the purpose of discussing this opportunity. I think the timing is right, and hopefully there will be some positive steps taken in order to allow us to generate rigorous data from single-arm trials that could potentially support MAA authorization. So I'll be starting things off, and then I'll pass it to a colleague from Merck in the US, Eric Rubin, to talk about a similar experience of how data from single-arm trials can be considered compelling in patients who have few other options. I'll be speaking to you about Crizotinib in patients with ROS1 positive non-small cell lung cancer, really stepping you through the background, the rationale, and the results that we've generated to date with this agent. So first, a little history. Not many of you may be familiar with ROS1 as a target and its relationship to lung cancer. It was first identified back in 1986, 30 years ago, as a cellular homologue of the avian sarcoma virus VROS. There was no ligand for wild-type ROS1 identified, and mice lacking this gene seemed to appear healthy. The cancer-related rearrangements in ROS1 were first identified in the human glioblastoma in 1987, but it wasn't until just 5 years ago, in 2011, that the first oncogenic rearrangements of ROS1 were identified in patients with non-small cell lung cancer. And so this was actually a very fortunate time, because during that time we were just beginning to develop Crizotinib. Large-scale screening of human non-small cell lung cancers found the frequency of ROS1 rearrangements in about 1.5% of tumors. So we agree that we think you'll agree that this qualifies as a rare abnormality in lung cancer. So the question in this situation is, how can a medicine be rigorously tested in such a rare circumstance? Here is the listing of the IC50 concentrations for Crizotinib, and as you can see, among the top 3 targets are c-Met, ALK, and ROS. And what many of you may not be familiar with is the fact that when Xalkori first entered clinical testing in 2007, it was entered as a c-Met inhibitor, because at that time we did not have the insights about the roles of either ROS1 or ALK in lung cancer, but yet we recognized that this was a drug that we could gain clinically meaningful concentrations that could potentially inhibit these activated tyrosine kinases. This is the preclinical activity of Crizotinib, and you could see the significant sensitivity in cell lines in which either ROS or ALK have been activated, compared to those that lack either of these activating kinases. This is a depiction of our phase 1 trial, Profile 1001, a trial that was initiated in 2006, and typical 3+3 dose escalation cohorts. And in this trial, as you may remember, 3 of 37 patients had objective responses, Those were molecularly characterized as we began to become aware of the rearrangement of ALK in a small percentage of lung cancer patients. And in fact, all 3 patients, 2 lung cancer, 1 sarcoma patient, each of the responders had rearrangement of the ALK gene. So we then began to explore it in molecularly identified subgroup of patients, those with lung cancer harboring the ALK rearrangement, and also in patients who had c-Met-positive tumors. But this trial, which has undergone, I think, upwards of 20 amendments over the past 10 years, then underwent an amendment to actually look at another group of patients in 2009, and those patients had ROS1 positive lung cancers, including non-small cell lung cancer. So in the next few slides, I will be showing you the evolution of data over time, because I think that this is something very important when we're talking about single-arm data. You don't have a concurrent contemporary control arm. You really have to evaluate this in the context of what you know about the disease. And that's where the strong signal will first become apparent. So that was in 2012 in a subset of 14 ROS1 patients. And as you can see here, in the 14 patients, there are 8 who had objective responses, a response rate of 57%. These appeared to be reasonably durable, almost approximately half a year, and a very high disease control rate. This was beginning to emerge as being promising compared to what we'd expect from chemotherapy in this group of patients, some of whom had had many lines of prior therapy. What also helped us in this evaluation was the context of Crizotinib in ALK-positive patients. So when you compare these results to those that were generated with a similarly small number of ALK-positive patients with a similar amount of follow-up, you see a very similar picture. There, the response rate was around 53%, and the disease control rate at 8 weeks was 79%. So by this point in time, we had much more experience with ALK, longer follow-up, and now we're beginning to see that pattern repeat itself in this rarer subgroup of patients carrying ROS1 translocations. And this is the waterfall plot of those first 14 patients. You can see very few patients were on the upside of this. The majority of patients had some element of tumor shrinkage. But admittedly, few numbers of patients, short follow-up. So these were interesting, provocative, worthy of further study, but by no means definitive. So here is, on an individual patient-by-patient basis, some of these responses were dramatic. And as you can see, this is a patient whose CT scan on the left. This is not pleural effusion. You can actually see the tiny air bronchograms in this mass of tumor in this patient. So the fact that 8 weeks later you have a virtually, I'm sorry, there's the CT scan that I was talking about. You can see that in the left lung is virtually replaced with tumor. The right lung is hazy, heavily involved. This patient was highly symptomatic from this disease. And on the right side, you can see within 8 weeks, there was virtual clearing of the disease, a dramatic response, albeit in a single patient, but again, another element that indicated that we had something here that was special, doing something much more than we would have expected chemotherapy or radiation therapy to be able to do. So now we followed these patients along. We expanded the number of sites that were accruing these patients, and we'll talk a little bit later about the molecular diagnostic that we used to identify this rare subset of patients. But by 10 months later, in April 2013, we had 36 patients on. And here, and what's a relatively unusual occurrence, as you gain more experience, the response rate doesn't go down and regress to the mean. It actually went up from about 56% to 61%, and we had 6% of those responses being complete responses. Waterfall plot, again, continued to show the vast majority of patients had some degree of tumor shrinkage as well. And here's the progression-free survival curve, which you see that at median progression-free survival is nowhere near being reached. The event-free probability of having a progression or no progression was 76% at 6 months. So you can see what we were identifying as 8 weeks or 12 weeks. Now we can have 6 months of follow-up and see that 3/4 of patients were without progression in this cohort of patients. And then fast forward another year, and these are the data that were published by Alice Shaw in the New England Journal of Medicine. This is the final group of 50 patients with ROS1 positive non-small cell lung cancer with, again, that very high response rate of 72% by investigator assessment, holding up very well, 6% complete responses. And now with the longer follow-up, we got a more accurate sense of the durability of this, which is another important element of a drug's activity. And the median duration of response was 17.6 months, median progression-free survival 19.2 months. So this was very compelling data. One of the questions that we had to ask with ROS1 was the same as we had asked with ALK positive non-small cell lung cancer. Had we simply identified a subgroup of patients whose disease had a good prognosis, that regardless of treatment, they would have done better than your average patient? And you can see here the answer is no. On the left side, in this publication by Mazieres and colleagues in 2015, you can see that progression-free survival with pemetrexed-based therapy was about 7.2 months. This included some first and second line patients. And the progression-free survival, this is relatively immature, was 9.1 months with Crizotinib, with some patients showing some longer-term freedom from progression. Here's the, what also happened was that we began to look at this in other studies as well. One study done here in Europe, the EUROS study by Mazieres, had 30 patients, overall response of 80%, very consistent with the Profile 1001 cohort, and here's the median progression-free survival of 9.1 months. And AXE, a study that was supported by INCA in France, in 37 patients, once again showing a 71% response rate. So we now had data from 3 independent groups showing response rates in the range of 60 to 70 to 80%, and the median progression-free survival with a low of 9, high of 19 months, and median overall survival is not achieved. And then a fourth study was done in Asia using a different assay, a PCR assay. This is the largest cohort reported as of yet. It was reported earlier this month at ASCO by Goto and colleagues, showing an overall response rate of 88%, median progression-free survival of more than a year, and overall survival probability at 1 year of 84.4%. Again, in a group that does not have a favorable prognosis compared to other non-small cell lung cancer, this 12-month overall survival rate is notable. Now, a word about the companion diagnostics that were used in the OxOnc study that was reported earlier this month. A PCR assay was used, central review was used to identify these patients. It was a single marker test. This is commercially available as a CE-IVD test and has been approved in China. But this also highlights one of the challenges for identifying these rare patients. If you have a one-off test where you have to have tissue sent to a laboratory and tested for one marker, in this case ROS1, in a small tumor sample, where you're also testing for other potentially actionable mutations, you run out of tissue, and this becomes a real challenge. So one of the things that, and this shows that in the OxOnc, and then actually in the profile study, we used fluorescence in situ hybridization as well. It was done primarily through a central lab, John I. Euphrates Laboratory at MGH. And there also, it was a single analyte test. But as we move forward, and now there are 10 or 12 potentially actionable mutations in non-small cell lung cancer, we have been working with regulatory agencies. We've been working with other pharma companies. We've been working with diagnostics companies to come up with a limited panel of genes, 52 genes, that may be relevant in non-small cell lung cancer, and therefore can use a multi-analyte NGS platform as a potential basis for more efficiently using the tissue to identify actionable mutations, and then use that as the potential basis for a companion diagnostic for regulatory purposes. So this is a work in progress. Our pharma partner in this is Novartis, and we've had a number of interactions with health authorities on moving this forward, because we think this moves the field forward not just for one company or one molecule, one asset, but actually the whole range of assets that are under development now in non-small cell lung cancer. So I think what helped us in understanding that we had something special with Xalkori in ROS1 non-small cell lung cancer was its similarity to what we saw with ALK-positive lung cancer. Similar disease, non-small cell lung cancer, similar activating genomic event in ALK and ROS, an event that was predictive to sensitivity of the drug, not prognostic. It was defined by a companion diagnostic with appropriate rigor and had high overall response rate, duration of response, and prolonged progression-free survival. And what you see here is the experience with crizotinib in ALK-positive non-small cell lung cancer from another phase 2 single-arm study. And this was actually followed up with a phase 3 study, one of 2 phase 3 studies done that confirmed the advantage of crizotinib over standard of care chemotherapy. In this case, second line. A subsequent trial showed first line chemotherapy, that crizotinib was advantageous to both. So having so many similarities and having a compelling case of a genomic driver in the case of ROS for this subset of patients, the question is, do we have sufficient similarity in these cases and evidence from phase 2 that we can say with some level of assurance that we have a drug that has a favorable benefit-risk relationship short of doing a formal phase 3 trial. So this is a summary of our interaction with the FDA, and you can see there it received breakthrough therapy designation in April 2015. The data package was 50-plus patients with ROS1 positive lung cancer from Profile 1001. There was the companion diagnostics, which was the FISH assay performed at MGH. The supplemental NDA was submitted in October of last year, and the supplemental NDA was granted in March of this year. So in summary, what I'd like to say is that the single-arm clinical trial data suggests that crizotinib has promising activity against ROS1 non-small cell lung cancer, that the clinical activity of crizotinib in patients with this disease appears to be similar as reflected by overall response rate, durability of response, and progression-free survival to what was achieved in patients with ALK-positive non-small cell lung cancer treated with crizotinib. Both the FDA and EMA have granted accelerated or conditional marketing approval to crizotinib in ALK-positive lung cancer based on the results of phase 2. The US FDA granted that in August of 2011 based solely on the results of phase 2 data. EMA followed a little over a year later, also basing its opinion on the phase 2 data, but only after the first of the 2 phase 3 trials had read out and they were aware of those results. So FDA has granted regular approval to crizotinib in March of 2016 for patients with ROS1 non-small cell lung cancer based on phase 2 data. And the question is, where certain criteria are met, are data from single-arm trials sufficient to support marketing authorization in the EU? And with this, I'd like to acknowledge many of my colleagues at Pfizer and elsewhere who helped me with this information. And with that, I'd like to end my comments and thank you for your attention.
So we have about a few minutes for questions. If you have any direct questions to Dr. Rothenberg. Professor Jonas Berg.
Thank you for this informative presentation. I think it will be of some relevance for the discussion onwards, but as I remember, you didn't at one single moment mention that drugs may have side effects, and that is of course a critical issue when one is evaluating drugs with this type of strategy. How do you know when you have compounds in for evaluation? How is the benefit-risk balance? And the risk is of course the side effect. And if you have targets where you have no major insight on the normal expression of that target, how will you have information with the type of studies, with the type of numbers, etc.? I'm not expecting you to answer, but that should be an important question to highlight during the day.
Absolutely, I agree that certainly regulatory action and use of any agent is based on a favorable benefit-risk relationship. For the interest of time, I was not able to go into those. Suffice to say that the safety profile was very similar in our ROS1 experience as it was in our ALK experience. There were no new safety signals. And now we have more than 5 years. We have more than 5 years of experience with Crizotinib on the market, and feel that that favorable safety benefit-risk relationship persists. In terms of the expression of the target in normal tissue, as preclinical data showed, there could be knockout of this gene, an untranslocated or rearranged gene, without any loss of murine viability. So while this does have some side effects, we don't think that this is related necessarily to the normal tissue being particularly sensitive. It really is the activated ROS and ALK genes that are the target and are most sensitive to this drug.
Professor Vassal.
Yeah, thank you. Gilles Vassal from Gustave Roussy. Thanks very much for this presentation, which exactly addressed the topic, because the drug was available, marketed in non-small cell lung cancer ALK positive tumors at the moment the information on ROS positive tumor was available. And then the key point that the development on one hand, and on the other hand, the off-label use of the drug by anyone who could prescribe this to patients without any further evidence. And I think this is a very critical point. Not only can we generate data and get to marketing approval with single-arm trial, but in addition, how to improve access to innovation early for this patient and gather the information. And you mentioned, in addition to the profile, the 3 other programs. One is retrospective collection of data prescribed in patient off-label. You mentioned the ACCES program, the National Cancer Institute program in France, which exactly addressed the topic. How can we give access to the molecular profiling of the patient, and then give secured access to the treatment for patients with positive tumor in a clinical trial that will generate information. And I think in our discussion today, it's not only generating evidence for marketing authorization, but it's also being sure that patients have access to innovation in a secure environment and we can collect the information. And it generates and the question of, can we get use of the information generated in the real world when the drug is prescribed in addition to the clinical trials?
Those are excellent points, and I agree. I hope we have a chance to discuss those more during the course of the day.
So if there are no other questions, we'll move on to the next speaker, Eric Rubin from Merck Research Laboratories on use of single-arm cohorts trials to demonstrate clinical benefit for breakthrough therapies.
Thanks, Professor Pignati. I'd like to thank you and Dr. Casale also for the opportunity to discuss this topic. And I'll focus on Pembrolizumab, our anti-PD-1 treatment. And I'll talk about two general areas. First will be Protocol 1, which was our first in-human study and is an example of the use of multiple expansion cohorts to generate evidence that was sufficient for regulatory decision-making. And then the second about ongoing single-arm trials with registrational intent and new indications with high unmet medical need. So Protocol 1 was a very interesting study. As I mentioned, it was a first-in-human study. It initiated in 2011 and was initially designed as a dose-finding study, typical biologics dose-finding study in oncology with a 3+3 dose escalation. And it had a single expansion cohort in melanoma with an estimated sample size originally of 32. There were striking responses observed initially during the dose finding stage in melanoma patients, and that led to an increase in the expansion cohort sample size to 60 and included both ipilimumab naive and ipilimumab treated patients. And I'm going to mention some of the statistics through this. There was careful statistical pre-specification during the multiple amendments that happened in this study, which there were a total of 9. And so the first one, there was a hypothesis around the melanoma patients that involved a 10% overall response rate and a 30% disease control rate in ipilimumab naive patients with an alternate hypothesis of 30% for response and 55% for DCR. using a Hochberg correction, and this had 97% power to exclude the null hypothesis with a one-sided p-value of 0.05. Because of the fact that IPI-naive patients had an effective alternative treatment, there was also a futility analysis that was included in the IPI-naive population. Second, there was addition of a previously treated non-small cell lung cancer patient cohort based on the suggestion that immunotherapies such as this drug could be active outside of traditionally immunoresponsive diseases such as melanoma and colon cancer. And so we added a 35-patient cohort of previously treated lung cancer patients, again with the hypothesis that involved a null overall response rate of 9% with an alternative of 22%, 80% power, and a one-sided p-value of 0.1. So with these data, as these data accumulated, again, we were seeing remarkable activity in both melanoma and then lung cancer. And it was around this time that we applied for breakthrough designation with the FDA. We were actually the first to get breakthrough designation in an oncology indication in melanoma. And that really enabled close discussions with the FDA around subsequent changes in the study that would again provide evidence needed for regulatory decision making. And so one of them was the definition of a ipilimumab refractory cohort. This is a very carefully defined population of patients who really were of high unmet need, given that alternatives in this situation were chemotherapy, which was relatively ineffective and toxic. So again, we had a statistical hypothesis there that I won't take you through. It's written on the slide. And then we also went through, there were some dose-finding questions, as there often are. And so much of the study involved randomized dose comparisons that involved comparisons of both the actual dose, so 2 milligrams per kilogram versus 10, and as well as a schedule question where we were looking at 10 milligram per kilogram given either every 3 weeks or every 2 weeks. And because of some uncertainty around whether dose might relate to tumor type, these were randomized cohorts in both melanoma and lung cancer. And to add further to the complexity of the study, our initial data in lung cancer suggested that most of the benefit there were in patients who expressed PD-L1 protein on their tumor cells. And so the study also contained training and validation sets to enable development of a companion diagnostic for PD-L1 in lung cancer patients. So again, these were all had pre-specified statistical hypotheses. We had multiple discussions with FDA enabled by this breakthrough designation in both melanoma and lung cancer. And ultimately, there were 1,200 patients that were treated in this first-in-human study with enrollment completed in July 2014. The schematic is shown here. Again, I'm not going to take you through all of the detail in here, but you can see that on the left is the original cohort A, which involved dose finding in various solid tumors. And then to the right, upper right is the large advanced non-small cell lung cancer cohort, around 550 patients there, again, including both various questions around dose and the training and validation sets related to the PD-L1 biomarker. And then at the bottom is the advanced melanoma population, over 600 patients there. Again, involved randomization to various doses and also involved questions related to both IPI-naive and IPI-refractory patients. So I don't have time to take you through all of the results of these studies, cohorts, but they were able to meet FDA requirements for accelerated approval in advanced melanoma and previously treated lung cancer. And in lung cancer, these were patients who expressed PD-L1 actually had greater than 50%, greater than equal to 50% expression of PD-L1 on their tumors, and these were both accelerated approvals. So just to comment on this approach, which I think was probably the first of its kind of such an approach in oncology, of using a first-in-human study to gain 2 approvals as well as a companion diagnostic. So there are benefits, of course. By using a single study, which was amended several times. One could efficiently address multiple hypotheses with appropriate type 1 error control, enabling evaluation of various populations, dose, and biomarker. Certainly was aligned with the use of single-arm trial designs as an accepted approach for seeking accelerated approval in the U.S. It could be performed with sufficient rigor to support regulatory filings. So we did, for example, use central independent review of imaging in this study. And it certainly accelerates development and approval for drugs that are transformative in nature based on early and strong efficacy signals. So this was clearly faster than if there were multiple separate trials that would in some sense involve replication of the early finding, and of course makes transformative therapies available to patients at an earliest opportunity, particularly in settings of high unmet need. However, there were challenges for this, and of course one is that the ultimate design was not predicted at study inception. I think there have been some discussions, including a recent paper from the FDA around this may not be something that would be appropriate to design for every first-in-human oncology study, to pre-specify a 1,200 patient study. And so again, we agree it would be difficult to use this approach for all new agents in a fully pre-specified manner, and it's not one that we use for every agent that we take to humans. There is a heavy operational burden on sites and the sponsor due to rapid accrual in multiple separate cohorts. And I'll note that there's also been discussion about, for example, adding pediatric cohorts to studies such as this. If you were to do that, this may require additional sites or investigators. I mentioned there were 9 amendments. This generates quite a bit of protocol complexity as well as potential issues with adherence to the protocol by sites. There was, of course, complexity of the analysis and interpretation of the data because of the multiple hypothesis testing that occurred simultaneously rather than sequentially. And so, for example, because of the concurrent development in both melanoma and lung cancer, we had to look at dose finding in both of those. Ultimately, the data have shown that dose really is independent of histology, but we had to do it in relatively large cohorts because we didn't know that a priori. And of course, you must ensure statistical rigor. I think there's some concern in large studies like this that there oftentimes are not pre-specified analyses where it's not clear how many patients would need to be treated before some decision was made. There were multiple database locks in a single ongoing study, also created programming complexity, especially around isolating a cohort for filing purposes, where again you have to lock and look at the AE rate, for example, at that particular time. And then while this was adequate for initial approval in the US, Canada, and Australia, it was not deemed sufficient in the EU, where randomized controlled data were expected to be provided before approval. So next I want to take you through some additional single-arm studies with pembrolizumab that also have registrational intent. These are ongoing studies in populations with high unmet medical need. And similar to the data that supported their approvals based upon Protocol 1, the idea here is that the risk-benefit could relate to analysis could relate to high response rates, in some cases related to underlying biology. And I'll give you examples of dramatic response rates seen in patients with relapsed refractory classical Hodgkin's disease. This relates to translocation in that disease that results in upregulation of both PD-L1 and PD-L2 protein expression. And then in mismatch repair deficient colorectal and other cancers where this genetic deficiency results in a very high mutational burden, which generates high sensitivity to checkpoint inhibitors, presumably as a result of neoantigen production, which can be recognized by the immune system as a result of this high mutational burden. And then finally, one of the other notable aspects of responses that are observed with pembrolizumab as well as other checkpoint inhibitors is the very durable effect that is seen in responses, presumably due to retraining of the immune system, where it can be difficult for cancer to escape this. And I'll give you an example of that in a study of squamous cell cancer of the head and neck. So with regard to classical Hodgkin's lymphoma, Keynote-87 is a multi-center, single-arm, non-randomized study of pembrolizumab given at 200 milligrams every 3 weeks, and involves 3 cohorts of patients with relapsed or refractory Hodgkin's disease. These include cohort 1. These are patients that have progressed after autologous stem cell transplantation and subsequent brentuximab vedotin therapy. There's a second cohort where patients have progressed after salvage chemotherapy and were ineligible for stem cell transplantation and also progressed after brentuximab. And then finally, a third cohort of patients who had a relapsed refractory disease after autologous stem cell transplantation but were not treated with brentuximab. So here are the results, response rates per investigator review from the 3 cohorts, as well as a cohort of patients with primary refractory disease. You can see that the numbers are relatively small, but in each case, there are high response rates in the range of 70 to 80%, with also a fairly high rate of complete remissions, around 30 to 35%, with very few patients having disease progression as their best response. This is also evident in waterfall plots, as shown here for the 3 cohorts. And then I'd like to move now to the microsatellite instability data. So this, the data I'll show you are from Protocol 16, a study done in collaboration with investigators at Johns Hopkins. The study design is shown here. It involved cohorts of both colorectal cancer patients, and these were patients that were either deficient in mismatch repair or proficient in mismatch repair, and then another cohort C that included patients who had mismatch repair deficiency in non-colorectal cancers. So you can see, and this is the colon data. The waterfall plot shows patients who were mismatch repair deficient, which were the green bars. You see, again, very dramatic activity with almost all the patients having tumor shrinkage. And in comparison, the patients who were mismatch repair proficient are shown in red. Most of those patients had tumor growth. If you look at response rates, the response rate in the mismatch repair deficient colorectal cohort was 57% compared to 0% in the MMR proficient cohort. Progression-free survival in the MMR deficient colorectal cohort was not reached, and the same was true for overall survival. Looking at diseases outside of colon cancer, a number that are shown here, the same type of effect was seen in, again, these are patients with mismatch repair deficient disease. Most patients had tumor shrinkage. There was an overall response rate of 53% in this population. So again, these both examples show that in these 2 patient populations, one can have a very high response rate on the basis, again, presumably of this underlying biology. So moving to squamous cell carcinoma of the head and neck, I'm going to show you data from Protocol 12, which was a single-arm, a study that involved single-arm cohorts of head and neck cancer, bladder cancer, triple-negative cancer, and gastric cancer. So these are patients with refractory head and neck cancer. The initial cohort was PD-L1 positive, but this was subsequently modified to an expansion cohort that included patients who did not require PD-L1 positivity for entrance. And the results that I'm going to show you involve combined analyses of both the initial and the expansion cohort. So in this case, we see that the overall response rate was perhaps not as dramatic as the other 2 cases I've shown you, 18% in this population. If you look at the waterfall plots, as is true when you look at, in many cases, at RECIST-defined response versus waterfalls, you do see that there's a higher population of patients who had reduction of measurable disease. So 60% of patients had a decrease in target lesions. But what's particularly notable here is the Kaplan-Meier at the bottom, which shows response duration. So you can see that in this cohort, median response duration has not been reached, with a range of 2 to 30 months. And again, as has been seen in other cancer types, the responses tend to be sustained, with 85% of responses lasting for more than 6 months and 71% lasting for 12 months. So in this case, an example of where we're not having these response rates of 50% or higher, but nevertheless very meaningful in this population, particularly with a sustained response duration. So talking about the benefits of approval based on single-arm trials, well, so as I've mentioned, for breakthrough immunotherapies, this allows earlier access of patients with high unmet medical need, as well as potentially rare tumor types. And one can ask, after with agents such as Pembrolizumab, where you've shown activity across multiple cancer types, whether randomized studies should continue to be done in every cancer type. And the feasibility of this could be questioned. I think, as has already been mentioned, one can ask whether single-arm studies could be done, could be the basis for an approval, and that these could be supported by use of real-world data after that approval. So talking specifically about challenges in the EU regulatory environment, and I'll thank my Merck regulatory colleagues for this slide. In the EU, the conditional marketing authorization scheme is currently limited to initial marketing authorization applications and is not applicable to type 2 variations, for example, for new indications. And this is in contrast to the FDA, which allows breakthrough designation and accelerated approval for new indications of an existing product, especially in the case of high unmet medical need and or orphan indications. And again, this may be a specific issue for immunotherapies that are expected to show efficacy in a large number of different tumor types. Thank you for your attention. I'm happy to take questions.
Thanks. Is there any question? Please.
Ken Mastris from Europa UMO, a patient. We mentioned in the earlier discussion about side effects, but the other thing is, from my point of view, what is the patient's consent in this sort of feeling and the quality of life for that patient? We haven't sort of discussed that.
Yeah, so similar to MACE, I didn't have time to get into the safety profile, but again, I can say that Pembrolizumab is generally well tolerated. There are some immune side effects. Happily, these tend to be relatively low grade and reversible, sometimes treated with steroids. However, there can be serious side effects. Pneumonitis is one in particular that we focus on. In our randomized studies, which have now read out, actually the side effect profile is better than chemotherapy. So that's been sustained by randomized data. And I would say it's the same for our quality of life assessments in those randomized studies.
Thank you. Aris Bamias from Athens University. I'm a medical oncologist. I'm here representing the GU subcommittee of the Educational Committee of ESMO. And I think that this example of this product today, these novel immunotherapies, is very relevant to what is going to be discussed in terms of their efficacy, but also the regulatory approval of these drugs. And I have followed this story in the bladder cancer, where these products, Pembrolizumab and atezolizumab, have been approved based by FDA based on the single-arm trials. And I think that this highlights actually the need to set specific criteria if one needs to move forward to that sort of regulatory approval. Because my feeling is, although they're they seem to be very effective agents, the single-arm trials actually raised some issues which my feeling is that they should be clarified through randomized controlled trials. For example, the safety issue, I don't think we know so many things yet about these novel immunotherapies. The way to assess response, we know the problem of pseudoprogression with these agents. And also, we still have a problem of selecting patients, the way to select it based on molecular evidence. So, and on the other hand, bladder cancer is not a rare disease. And in fact, phase 3 trials are accruing very quickly, much more quickly than they thought. So results should be there. So I just think that we need to set specific criteria where single-arm trials should be the basis of regulatory approval.
Yeah, I would agree. I think this is a very critical discussion point. I mean, my colleague Roger Dancy is with me, who leads the development of Keytruda, and may want to comment on our bladder cancer approach. But we have both single-arm studies as well as randomized studies, and I think in larger patient populations, this can be done. Although I do think that as time moves on, with the increasing availability of these agents, the ability to have non-confounded, for example, overall survival endpoints is going to be difficult. And so we will have to consider other endpoints. I know we'll also get to the HTA discussion a little bit later. I do think this is an issue, even in diseases which are not rare.
Jonas Berg-Karlinski again. You mentioned different areas of indication and different areas of exploration. When you really are looking at the data, then of course one wonder what is identifying the patient who have gained, respectively no gain of the installed therapy. And one thing which has been discussed to some extent is the mutational load, presence of passenger mutation and creation of neoantigens, etc. And you just mentioned a simple thing like target, PD-1, PD-L1, etc. And the more I look into those data, the more confused I get if I look in between different drugs and different studies. So what have been your strategies? Because the society in the world would like to, whom are the patients to receive these compounds? Because everyone can't have those compounds for economical reasons presently. So what have been your strategies? Because you will be asked these questions for sure by some agencies. And how are you thinking around this?
Yes, this is a very, very important question. And I think that, we've looked at it a number of ways. When we first started the trial, the only data available really in the field at the time in terms of trying to understand could we identify characteristics that would identify patients most likely to respond was PD-L1, one of the ligands of protein expression. And so that was something we looked at early on in our trials. And as I mentioned, particularly in lung cancer, this seemed to be predictive, and where patients who didn't have that expression did not seem to have the same degree of benefit. And that was unlike our data in melanoma, where most of the patients with melanoma were actually PD-L1 positive. It didn't look like going down the path of selecting patients just for treatment on the basis of that expression would be meaningful. And so we went through a path of where we looked specifically at PD-L1 positive patients in our both initial filing, as I mentioned, and even in our subsequent randomized studies. We can probably spend some time talking about this. I think it's clear that PD-L1 is a good test. I like to say many tests are useful, but no test is perfect. And so it's certainly the case here, as is true for other IHC tests such as HER2. So it's not perfect, right? We and others can find patients who test negative and yet will respond. It's not a high percentage, but it does exist. So it gets into discussion, well, how can you prevent then every patient having access to the drug when we can't say with 100% certainty that that patient's not going to benefit? Yet we can say, we and others in randomized studies, that the majority of the benefit really is in the positive patient population. I'll add that we are also heavily invested in additional work to identify patients who can respond. So we have an RNA signature that we're developing, potentially towards a companion diagnostic, as well as work in the DNA space, as I mentioned, around MSI and even in a mutational burden assay. So ultimately, right now, of course, it's very confusing, right? We don't have the perfect biomarker. We've got PD-L1 tests that are approved. There's additional complexity there because there are multiple tests that are approved at this point. Perhaps in the future we'll get to tests that will be closer to perfection, where we'll have more confidence. We can tell someone that if they're test negative, the chances of response are remote. But until that time, I think this question is going to come up.
A short remark. You are talking positively about PD-L1 or PD-1 testing, but I must say, you know, there are several companies out there, and I must say they have different cut-off, different tests. And,
Yes.
but when I sit with the data, I'm sorry, my confidence in that antigen for predicting response is quite limited, contradictory to your strong view on this.
Fair enough. Yep.
Ignacio Garcia from Takeda. So based on the complexity of the design and the multiple issues in terms of data review and controlled implementation of the trial, to what extent do you think that this design is something to repeat in the future, or if it is something that just happened at one time? So can we follow this for other drugs?
Well, I would, again, we can talk about this. I think for something that has transformational aspects to it, as this one did, I would think we should. Again, I think that it gets very effective treatments out to patients as rapidly as possible. There's no doubt that there are patients who, I think, without access to the drug in a rapid way, would have died. So I think that, but the question that I think Professor Pignati raised is, how do you define transformational? I think is the tricky part, right?
I'm Paul Oregon from University of Manchester Christie Hospital, and I'm representing ESMO Melanoma Group. So I think the Pembrolizumab story is a beautiful story, and I think immunotherapy is absolutely critical to the discussion today. But melanoma, lung cancer, bladder cancer, Hodgkin's disease are not uncommon cancers, and what we're talking here is about early approval and subsequent verification of that with randomized studies. But there's another group of trials of patients out there where they're very rare cancers and where it's not at all clear whether immunotherapy will work or not. And I think we need to focus on the trial design for that group of patients. And I think the trials we need to be confident based on a phase 2 study that the drug works, but also confident in many indications that it doesn't work, because there's a huge expectation for immunotherapy which is not completely founded that it will work in all cancers.
Right.
And I'll give an example of that. In melanoma, clearly pembrolizumab and nivolumab are licensed in melanoma, and they're licensed in the first line setting. But in fact, the evidence that they work in subtypes of melanoma, such as mucosal melanoma and ocular melanoma, is very poor. And yet, those drugs are now licensed and will be used forever until a better treatment comes along. So we need to have trial design that picks up ineffective drugs, but also in rare populations, and that is a real challenge.
Colleagues, why don't we hold on to these very important comments until the discussion. Paolo, over to you with uncontrolled evidence in rare cancer.
Thank you. So the problem is why randomized trials may not be the best thing to do sometimes in rare cancers. Assuming we have a definition, and you know that clearly there are a lot of problems on this, as long as of course melanoma is not rare, but then you go to advanced melanoma and then you go and so on. So clearly the problems are on the table, and you are very well aware of all this. When we published this first attempt of Rare Cancers Europe to get to a consensus on the methodology of research in rare cancers, we tried to address some of these things also in regard to randomized trials. And I remember we were here the day the paper was published on the Annals of Oncology, and I must thank EMA again for this. And we decided to have meetings thereafter, especially on very rare cancers. And the first one, I hope, the first, was on chordomas. And this was the tweet of the Chordoma Foundation that very day. They say they said progress. They were very enthusiastic about the meeting we had here that day. And the feeling was that regulators, patients, and clinicians were on the same side. Because we touched the problem of a very rare cancer in which clearly randomized studies are by definition difficult. Also, clearly one could say, okay, but you could also do a small randomized study, a low-powered randomized study or something like that. So the problem is, should we choose controlled evidence or uncontrolled evidence aside from the problem of numbers? So not necessarily the number defines the... And I remember that one of the suggestions which EMA gave to the Chordoma Foundation was why don't you establish a clinical database on your disease throughout the world in order to generate historical controls, which then could be used when there will be some drug in chordomas. And by the way, the Chordoma Foundation decided to do this. So they decided to establish a clinical database. So picking up your suggestion and hoping in new drugs. Of course, the problem here is that now with electronic patient records, we will have the possibility to interconnect them, so to generate almost automatically this big data, big clinical data. We always talk about big data, about genomics and so on, but there are also big clinical data. There will be more and more with the interconnection of electronic patient records. And the methodological challenges implied are formidable. I think we tried to spend at least a paragraph in our paper on this, because clearly the selection bias of this kind of observational evidence is a challenge. And so I see here a window of opportunity for our discussions, because it is an opportunity, it's also a challenge, how to use this big data in the near future, not only for regulatory purposes, but also for regulatory purposes. I mean, for advancing our knowledge. And another thing we said in the paper was that it's not the study design which defines the stage of development and vice versa. So I mean, it's not the uncontrolled nature of a study which defines that study as a phase 2 study, if our questions are phase 3 questions. So if we are interested in survival, we are interested in the final prognosis of the patient. Not necessarily an uncontrolled study is a phase 2 study. Sometimes we see the vocabulary of phase 2 studies used even in the statistical plans of the studies, just because the study was uncontrolled, because the decision was that a randomized study was unfeasible. You must be aware that it's true that lung cancer is becoming a set of rare diseases because you split up lung cancer, but we must also be aware that the same applies to rare cancers. So rare cancers are split up as well, so they become even more rare than before. And I would, since I follow sarcomas, I would just like to give you a couple of examples from the sarcoma world, which is made up, of 50 histologies at least. So we have a vexata question in sarcomas, whether a multi-agent chemotherapy is better than a single agent. We have randomized trials, and I remember that when we randomized trials which point to some difference, but not so much. And in our guidelines, you will read that standard therapy is single agent, but multi-agent chemotherapy may be used, and in some histologies, you may have other drugs depending on, based on uncontrolled evidence and so on. I also remember that Paolo Bruzzi, who is here, joined our consensus meeting, the ESMO consensus meeting on sarcomas the last time, and he told us it's very strange because the only discussions you had, the only controversies you had to do with issues which were covered by randomized trials. So you didn't discuss on the other topics for which only uncontrolled evidence was available, but because the randomized trial is not necessarily the last answer to our questions. And I remember that the European sarcoma community appealed to EMA when trabectedin was rejected, was not approved on the basis of uncontrolled studies. And then it was approved on the basis of this controlled study of trabectedin versus itself. And the FDA approved trabectedin thereafter on the basis of another randomized study versus dacarbazine, without a survival improvement, but a progression-free survival improvement. I mean, in that story, it was clear that from the regulatory point of view, the randomized evidence was needed, because otherwise what we said, that trabectedin was working in sarcomas, was not believed in. But it's also true that when we went to review the whole compassionate series of trabectedin in sarcomas, we discovered that it is effective in myxoid liposarcoma, which is a subgroup of liposarcoma, and specifically in liposarcoma in a completely different way. And the study databases didn't allow us to conclude this. Only the compassionate series, and I remember that here at EMA there was some, some discussion about the wide compassionate use, which was done at the time by the company. But really, this series allowed us then to understand that even the mechanism of action of trabectedin is different in myxoid lipo, because there is a chromosomal translocation, so the fusion transcript targets some genes, and the drug displaces the fusion transcript from these genes. But I mean, we wouldn't have understood all this without in kind of uncontrolled evidence, of low quality uncontrolled evidence, which by definition seems to be the compassionate use of a drug. Then pazopanib was approved in soft tissue sarcomas on the basis of this progression-free survival difference, but then in all sarcomas, with the exception of lipo, because the study was not done in lipo sarcoma. But then the problem is to go to the subgroup analysis and try to understand which subgroups are targeted by this drug. And then we have preclinical data and uncontrolled evidence on several drugs which are similar to pazopanib. Our feeling is that these drugs work differently across histologies, but clearly we cannot do large studies on each of these histologies. We have uncontrolled evidence that the response rate may be substantially different across these drugs. All of these drugs, with the exception of pazopanib, are not available because they have not been developed, because companies clearly didn't believe that, that they could afford developing in these subgroups. This was the decision of companies, but clearly this reflects probably the regulatory implications. And so we cannot, we have uncontrolled studies in which we have several patients with a single histology, and the drug is not developed, is not approved. we have randomized studies in which these histologies are probably not represented at all, so not a single patient, but the drug is approved also for that histology just because it was included in the, it fit the inclusion criteria of the randomized study. So clearly this is quite paradoxical, I mean, in principle. And this, all this results in a huge off-label use of drugs. I remember when I came here with the ESMO position paper on the off-label use of drugs in Europe, you told me, okay, but we are the European regulators and you come here talking about something illegal. And it's true. But now I know that EMA is involved in a survey on the off-label use of drugs. But I mean, the regulatory consequences in practice of your decisions will be a high level of off-label use, which in Europe is really problematic, you know, across Europe. As a matter of fact, there are some European countries in which more or less you can use drugs off-label. There are other countries, EU countries, in which you cannot. And so this results into discrepancies in the access of patients to care. And at the last ASCO meeting, I discussed a poster of the French group pointing to a survival in leiomyosarcoma in their clinical database on all their patients, which was quite striking. And there are other data like this. You know, most probably this is due to the fact that in soft tissues, in leiomyosarcoma, we can use several agents sequentially, some of them even as a maintenance therapy. Clearly, if you look for a survival benefit in each of these lines with a study, even with a randomized study in each of these lines, probably you will not see any survival benefit, even because you will have any kind of crossover in clinical practice. Only if you did a study on an approach like this versus, I don't know, placebo, which clearly is a study which is unfeasible, probably only in this way you would see a survival benefit. This is our problem, and it's difficult to say how big clinical data may help us overcoming the problem of. But at the same time, I understand very well what was already said, that we could also generate data by allowing the use of the drug provisionally, and this has to do with adaptive licensing or so, I mean, or things like this. Clearly, we would be very much in favor in principle to something like that, because this allows patients to have access to the drug, and at the same time this generates evidence for the future. It has already been said as well that in the end, what is convincing for clinicians? I mean, in the last analysis, it's something like seeing a tumor response, some kind of tumor response, which lasts for a while. This is what convinces clinicians. Maybe we are wrong. Clearly, as we stated in the paper, validating a surrogate endpoint is difficult in rare cancers because you would need numbers, and the numbers are exactly what is lacking in rare cancers by definition. So validating a surrogate endpoint like tumor response is not easy. On the other side, if you have this patient with this coat compression, it's very clear that if you achieve a response, this patient will have some benefit, at least in quality of life. If this patient with a rectal gist undergoes some shrinkage of its tumor and may be operated on conservatively, there will be some benefit in quality of life. If this patient undergoes some response so that the mesenteric vessels release the tumor, which becomes operable, clearly there will be some increase in its chances of surviving. So in the individual patient, you may also state which is the proportion of response which is needed. So you may get to a personalized criterion of response for that patient in order to have some improvements in his chances of getting some. So you already showed the slide of the parachute, but this means reasoning in clinical medicine. So, so combining what we know with what we can infer logically in the single patient. And you know, which is the problem about tumor response? There are many problems. In the sarcoma field, we learned many years ago that tumor response may look like this. So it's not necessarily only tumor shrinkage. This gave rise to a lot of placebo-controlled studies just because it was felt that an external review was needed because tumor response and progression, of course, as well might be subjective. We developed some criteria for response, but what is the problem? The problem is that the subjectivity in assessment may increase. And all the more, I mean, now with immune therapies, we have other problems with tumor response assessment. I understand that the medical oncology community, unfortunately, has made a few steps forward following the first definition of response by one of the fathers of medical oncology. It was David Karnowski in this paper. And from then on, the tumor response assessment has always been about reproducibility, because the reasoning was it's useful for phase 2 studies, then it will be phase 3 studies which will tell us if the drug is effective or not. So our concern with tumor response is just reproducibility, which you know maybe is the same by definition in these two cases. So reproducibility is not everything, but all the literature on tumor response assessment in medical oncology is always also our criteria. I mean, you will always read that tumor response is not for the clinic, is just for being reproducible in the phase 2 setting. And this has been a mistake, because now that we could need a clinically sound response concept, we don't have it. In other words, we should try to have a tumor response fit to be personalized on the single patient, a tumor response assessment which clearly should be done on the tumor volume and not on something too simple. Non-dimensional changes should be incorporated. Some temporal dimension should be incorporated in the concept, because clearly a tumor response lasting 1 week doesn't mean anything. Tumor response is more and more seen as a continuous variable. I mean, when you use the waterfall plots, practically you use tumor response as a continuous variable. Probably even the growth rate is important. Sometimes even is slow, kind of slowing down of the tumor progression may be meaningful clinically. You understand that all this is difficult. So when you told us once, I remember, we are not necessarily in favor only of RECIST criteria. If you come up with new criteria which have been validated, which are sound clinically speaking and so on, it's fine. The problem is that it's not that easy. I think that this is a second window of opportunity and of challenges for us to develop a new concept of response, because clearly we need it. All the attempts which have been done have several limitations, but clearly we must go beyond reproducibility. We should go towards the clinical meaning of tumor response. I already told before that that ESMO has embarked into an effort on the magnitude of clinical benefit. I mean, this means that our professional societies, scientific societies, are more and more willing to be engaged in these methodological issues. And so we are. The problem is that in rare cancers, we must accept a degree of uncertainty. We must accept an extra degree of uncertainty because otherwise rare cancer patients are discriminated against. So there might be some more subjectivity in a new tumor response definition, for example. But this should be accepted, I think. In a sense, as a clinician working on rare cancers, I would say to regulators, let's not make compromises on the magnitude of the benefit, because 1 week is 1 week, independent of whether the tumor is frequent or rare. So the same benefit should be asked for in rare as in frequent cancers, but the degree of uncertainty should be inevitably be higher in rare cancers. Thanks.
Thank you very much, Paolo. Before we open for discussion, I take my chairman's prerogative to ask you a question. I find it's very interesting that you want to look beyond beyond conventional criteria and to almost individualize response and try to learn if there is a benefit or not. At the same time, if I perhaps an initial reading of the ESMO guideline, as you mentioned before, is almost trying to raise the bar to single out the exceptional cases, and I know the objective of that piece of work is perhaps precisely that, to single out the very, very exceptional cases. But does this range of approaches really reflect actually a rather heterogeneous view on all these matters within the clinical oncology community of what matters to patients? Is it still so polarized that there are this is all about overall survival, the rest is, soft endpoints which are probably meaningless, and the very sort of progressive exploratory type of view? How big is this debate? I remember, I think it was ESMO a couple of years ago, Jonas, you must remember it as well. We have had a debate with a vote with ESMO participants on PFS versus OS. Clearly a wrong type of question, but just to see how polarized views were, and they were kind of 50%. So, and at the same time, industry, when you come to regulators, and sometimes you say, okay, phase 3 is required here or not, is this just reflecting the same, if you want, broader uncertainty that people have, attitudes to uncertainty? But actually, it's never very clear to me how these skepticism or whatever progressiveness are justified. So how homogeneous are you? How homogeneous are the regulators? And are we improving?
Because I go back to this slide to answer your question. Because if you don't split the magnitude of the benefit from the quality of evidence, you may believe that there is a problem in the oncology community. Because sometimes you say, okay, let's accept tumor response as an endpoint, and that's it, and so on. The problem is simply that when I say that a significant tumor response lasting for long has a clinical meaning, I imply that most probably there will be a big survival improvement. The only problem is how I can assess that survival improvement if the numbers are small, if the study designs are challenging, if you had to consider several lines of therapy, and so on. So I'm not saying that I don't want to raise the bar. Raising the bar means that 1 week benefit in survival doesn't mean anything. And I think that the oncology community today is quite in agreement on this. Of course, if 1 week is a statistical indicator by which there are a few patients who survive forever, and because they are cured, okay, clearly this is another issue. But this is, again, a methodological issue. You must just, be able to pick up this benefit in a few patients, but in the end, they will have a long survival. So again, there is not a contradiction, I think, between saying, let's look at the quality of evidence in innovative ways. And in return for that, I say to you, the magnitude of benefit should be high enough as to justify that treatment. So I don't see frankly, any contradiction in this.
So thanks, very provocative question. I think that actually clinical trialists, whether you're talking about cooperative groups, academicians, or companies, have been quite creative and quite responsive in terms of identifying and defining endpoints in clinical trials. They're not static, although we may think of them in that way. So one of the things we always have to think of is what is going to be the meaning and impact of a new therapy on patients? How is it going to be perceived to be of benefit from physicians who treat the disease, to the payers, and to regulators? So those things have to really be in our mind. But I've felt throughout my career that it's incumbent upon us, when we see a signal of activity of a drug, to be able to tailor the development plan, including the endpoint selection, size of the trial, et cetera, on the unique characteristics that we're seeing emerge from those clinical data. So actually, I could refer back to experience that I've had 20 years ago in the development of gemcitabine. In the design of that trial, we were beginning to see something that we hadn't seen before. Not only tumor shrinkage in patients with pancreatic cancer, but alleviation of some pretty debilitating tumor-related symptoms. Now, this is in an era before the standards for collecting patient-reported outcomes and health technology assessments were as high as they are today. And we created a new scale, and we called it clinical benefit response rate, that measured pain and weight loss and performance status. And actually, as you know, the phase 3 trial showed that there was a slight improvement in survival and a significant improvement in clinical benefit response compared to standard of care chemotherapy. And I think had that trial been designed with only one endpoint but not the other, the drug never would have been approved. It was really compelling evidence that came forward at a time that it was accepted, and it ended up being a cornerstone for therapy over the years. So I think that's really the key, is being able to recognize and keep up with the very dynamic environment we live in cancer drug development. What we are seeing is that more and more diseases are being characterized beyond just their histologic appearance, but their molecular characteristics and now the immune infiltrate as well. This is creating opportunities but also challenges, because as was referred to, lung cancer is no longer divided into small cell and non-small cell lung cancer, but it's based on the molecular characteristics and now the immune profile as well. But I think we should take advantage of that. I don't think that we should be considered ourselves victims of technology and that we identify even smaller and smaller subgroups of patients, but we should find ways of being the beneficiaries of that greater insight.
Ken Pastris, Europa UMO. Welcome Paolo's presentation, and as patients we do need big data to support our patients. But just an observation as a patient, where is the patient in the actual trials? It should be, the patient should be involved at the beginning. EMA and ESMO have got patient advocates. Do we have them on the NFA, the HTAs and places like that? Just observation there.
Christian Dietrich, I'm on the ESMO side, responsible for the principle of clinical trials and therapy group. I'm glad about the situation. I think we're all aware that we have to treat rare cancers differently, also from the regulatory perspective than entities which are still to be called big entities. But I see a certain danger if we are taking this progress in dealing with the subject to an area where this is not absolutely necessary. Before the situation of, for instance, of bladder cancer and the atezolizumab situation was addressed, where a trial with more than 300 patients in a just phase 1 was done, and one could have easily done also a prospective randomized phase 2 in such a case. So if this happens, this lowering the bar from the registration point of view, then because all of those drugs which are now going to be developed in the future start to be, let's say, 10,000 euro per month, a second level will introduce to decide upon whether the population will get it or not, which will on the level of the HTA. And I doubt that the health technology assessment frame should be the one taking over then the final decision. I would feel better if, let's say, the most scientific based approach and decision still is the one which is taken by the regulators, and not going to be shifted for, let's say, socioeconomic reasons to a pseudo next level.
Lothar Bergmann, University of Frankfurt. I'm a little bit skeptical about the relevance of response for an early approval of a new drug. I think it depends on the intention of the therapy. Is it with a curative intention or is it on a palliative intention? In a palliative setting, I think we must ask what are the relevant endpoints for the patients. And for the patients are the relevant endpoints symptom controls on the one side, and on the other side, a prolongation of overall survival or a less toxic therapy compared to standard therapy. And we cannot use the response in general as a surrogate marker for overall survival. And it differs from entity to entity. We have to show that it's really a surrogate marker. And, and additionally, if we are looking for the new immunotherapeutic approaches, we frequently see no benefit, real benefit in response, in tumor shrinkage. We even don't see sometimes a benefit in progression-free survival, but we see a benefit in overall survival. So I think we have to differentiate a little bit about the intention of the therapy we are doing. And I think this is important also to balance between the risk and benefit for the patient and for the approval as well.
Just to specify that when I talked about response, I was talking about whichever response. So it's difficult that something doesn't happen to the tumor in order to get a benefit. The problem is that it's not necessarily tumor shrinkage. As I said, we need new tumor response criteria, but I was very broad in the in defining tumor response just for.
I'm Rolf Steil. I want to go back to the first two presentations, and because I seem that to me so much enlightening, and I was wondering whether we couldn't, we go from things that are quite certain to very uncertain. But the first two, I think the example that there's a drug approved in one indication and the side effects are known, and now is, do we need a randomized trial or please not for another indication where there is very plausible evidence that the drug is as effective as in the original indication? And I was thinking, this is so clear to me, and because I think now the discussion is drifting to, issues which are very difficult, whether we could have a consensus on the first two presentations that this would be really an area where it's very clear that you do not need a randomized trial.
So I wanted to go back to that.
Thanks. I think it's an excellent point. And but is it very clear? Was it very clear? I guess your experiences during the evaluation is that there were times where this wasn't very clear. And in fact, you had a situation where these were lung cancer patients that otherwise would have been treated with standard chemotherapy. And did you have a good external control, so to speak, to say in the ALK positive patient, this would have been the survival on chemotherapy rather than not? And did you ever ask yourself, should I actually be doing a small randomized controlled trial here?
Right, very good questions. And remember that with Crizotinib, we have done 2 randomized trials against standard of care chemotherapy in the first and second line settings, and in both there was a statistically and clinically meaningful improvement with the use of Crizotinib. But I think that the point is, when do you know you have something special? Because so many drugs have a glimmer of activity early on, and maybe 1 or 2 dramatic responses, but the majority of patients don't respond. There might be certain toxicities that we have to be concerned about. So it really has to do with the duration of the follow-up and the numbers of patients. So that's why in my presentation I really told the story of what the early data looked like, because our interactions with physicians and with regulators was initially very interesting, but come back later when you could tell us more about the durability of those responses and whether these results hold up, or is there regression to the mean and you see a fall in the response rate. And in fact, with additional follow-up and additional patients and several independent trials, the results have been remarkably consistent. And when you look at not only the response rate, but the durability of those responses, that also translates into a very important clinical benefit. The other thing that really stands out with me and with many physicians I've spoken to who have treated patients with Crizotinib for either ALK or ROS altered lung cancers is the rapidity of response. I showed a CT scan of a patient who was obviously very sick with their lung cancer. Many of these patients have chest pain, cough, severe shortness of breath. And the stories about patients who start this medicine, which is a pill taken twice a day, say that in weeks and sometimes days after beginning therapy, they're beginning to feel better. And this, we don't think, given the CT scan, is a placebo effect. So it really is understanding the patient beyond just the usual response rate criterion of X or Y, but it's understanding it in the context of the larger benefit picture, measuring all those other elements, including the patient, as well as looking at the benefit versus risk and adverse event profile. I think once you look at it from that perspective, the picture becomes clear.
For me, the case of Crizotinib in ROS1 is a best case scenario. And something that has not been discussed is that you were with a safety database of probably 1,500 patients with the ALK positive treated patients. I wonder if it would be possible for regulators to accept this efficacy data in non-small cell lung cancer that are very good for any clinicians really showing that there is something there, with just the database of safety database just coming from 50 patients. Would it be possible to approve this?
We have a number of regulators in the room. I don't want to be the only one. We don't have any fixed number, and the current approach is to say, well, let's look at those 50 patients and look at the unmet need and who was going to make the decision of taking the risk of the unknown safety profile, and it's complex. But I think the experience is that our requirements are often totally overinterpreted. And then, when we just express a little bit of skepticism about, well, ideally here we should have a randomized controlled trial, this is taken by companies, we absolutely now cannot do anything else. And so I think there isn't a clear message from us on this point.
I just want to go back to what I said and maybe a little better explain. I meant these 2 situations you have to look at in the context. For the one, The context is we know an oncogenic driver mutation is there. And of course, this is very particular. I'm very happy to be in this field in lung cancer, where several of these mutations have been identified. So you know what second-line chemotherapy does provide, and you know the potential of an oncogenic driver targeting, which I think has to also put in the picture deciding on allowing or not a straight phase 2 study, plus the quite clear difference in toxicity. And this is regards the Crizotinib example, but also the Pembrolizumab example, versus the toxicity of a standard treatment. I can only talk now for lung cancer. So I think this is also part which has to be taken in context.
Can I? Okay. I think it's what is coming for following what Rolf you were saying, Giorgio Massimini from Merck KGaA. One thing that is not coming in this discussion at the moment is pharmacodynamic. If there is a target, the drug is a target that has an effect, and this is specifically true in rare diseases where you have a specific oncogene or a specific situation at the molecular level, this is a convincing evidence that, at least in my opinion, that you have something there that is worth pursuing. If you don't see anything when the target is not present, is also a convincing evidence that it doesn't work. I don't know if you did this kind of experiment in the Crizotinib ROS1 development, but I think you did. And this can be a very important information to understand if really you have something that is working in that disease in this particular situation or not. In situation like immuno checkpoint inhibitor right now, the problem is that we have not identified the target. The MSI must be 1. We don't know if this is 1, but it's in a sense our insufficiency in identifying the correct target in that particular situation that creates the uncertainty that has been discussed before. With regard to polarization, if you allow me a comment, it's not polarization, it's dialectic. We always discuss this kind of stuff, and there has never been 100% concordance of opinion on anything. I think we will have to live with that.
Well, when I go to surgical meetings, I hear surgeons presenting cases that went well. They were very smart, everything went well. And this morning we heard 2 histories of successes. But I think that there are many more failures, early signs, positive signs that were not confirmed either in phase 2 or in phase 3 randomized trials. We should be aware that without a control group, it is very difficult to assess the effects of treatments, also in terms of cost-benefit. When we look at the ALK positive data, in the randomized trial, the control group goes much better than we were used to see in non-small lung cancer, non-small cell lung cancer. So we should be very careful before stating that these 2 cases are paradigmatic of what occurs in cancer, clinical cancer research of new drugs.
I want to take you further and argue against what you say. These 2 cases are paradigmatic because I think we would like to show that a single-arm trial may lead to approval because the data has been exceptionally high. There may be single-arm trials where the data is not good, and this will not lead to approval. So, but the 2 examples were really a drug has an indication, the toxicity profile is known, and there's a rationale. One is molecular, the other is more difficult with immunotherapy to use it. I mean, of course, this is a very special situation. It doesn't apply to all situations.
In the magnitude of clinical benefit scale development, and we did extensive field testing before we published the first version. And what was quite obvious by faculty testing, but also by going to patients, is that people find it very important when they grade a drug, what is the toxicity, what is the burdensome side effects for patients, so having headache for a year, even though it's not that intensive, is a terrible side effect, and the quality of life. So if we are moving more towards single-arm studies as registration studies, then it may be very important if also patients are going to play a role in how important a drug is in the end for use in the clinic, to consider side effect issues, serious toxicity issues, and quality of life. And that's an additional challenge in single-arm studies.
May I? So I am Nicola Fazio, I'm a medical oncologist, and I'm particularly involved in neuroendocrine tumors. Therefore, this is a rare tumor field, as you know. My opinion is that in rare tumors, phase 3 are not so representative of the real-world practice that we usually have in front of us. But once we told in NET, we told that it was impossible to perform a phase 3 trial due to the high number of patients that should be enrolled. Now we have several drugs approved on the basis of phase 3 trials, even with a large number of patients. The problem is that we have the possibility now to use drugs approved on the basis of phase 3 trials where the comparator was difficult to be defined. Sometimes the comparator was the placebo arm, but in real practice we could use several therapies outside the clinical trials. Some other times the comparator, as for example in the last regulatory trial of PRRT, is a high-dose somatostatin analog that was never validated before, and this was a design of a regulatory trial. Therefore, there is a sort of bias in the comparator arm, in the control arm, and therefore it's difficult to apply that approval drug in clinical practice. But on the other hand, we have the possibility to use some therapies outside clinical trials never approved on this same basis. An example is chemotherapy. You know that chemotherapy in NET was approved since the 70s, streptozotocin-based chemotherapy, and it was never based on no randomized trial, neither randomized nor phase 2 prospective trial, but it was based just on retrospective data. Therefore, there is a sort of confusion in our clinical practice, and just for that, I think that in rare tumors, specifically in NET, could be better to move to the phase 2 single-arm trials. But of course, I totally agree that for a regulatory phase 2 single-arm trial, we should be sure about the clinical setting that we study, and just for that I would like to know which are the criteria that, for example, the regulatory agency like EMA has to be sure about the real need of that clinical setting. I totally agree also about the endpoint. The response rate couldn't be the only endpoint, but of course it's better to think about a combined endpoint, response rate plus duration of response, but plus also other clinical endpoints, some clinical benefit endpoint. Therefore, my issue is that phase 3 are not representative for rare tumors, and it's better to have a phase 2. And of course, we are working to find the most objective criteria and homogeneous criteria to define the real phase 2 in this field. But my warning is that in the meantime, we can use several even toxic therapies without any evidence and without any limitations in our clinical practice. Thank you.
Yeah, I think in our discussion today, it is very important to separate two very different scenarios. One is the scenario of really very rare disease, which do exist, where randomized trials are just impossible to do. I'm not talking about NETs because, as Nicola said, it's feasible. But I'm talking about, chordomas or Merkel cell carcinomas or other really specific tumor types that, it's just not realistic to ask for randomized control data. And the other, so in that scenario, I think we have no choice. We have to evaluate lower quality data to try to come up with an opinion. But the other major thing, which is totally different, is in a scenario where a randomized trial is feasible, but we are facing a really breakthrough drug that has a major impact on clinical outcomes. And I think that is the case, for example, for Crizotinib in ALK or ROS positive non-small cell lung cancer, particularly in a setting where we know a lot about safety issues of the drug. So maybe it was wise to ask it for ALK positive in the beginning, where we didn't know a lot about the quality of the responses, durability, impact on more long-term endpoints, or even security. But now, it's even a low-risk drug with a favorable safety profile. And, we are talking about 70–80% major response rate that are deep, that are prolonged. I think this is a very different scenario where we need to question whether we should keep our patients several years from being able to access this drug that has a major impact on their outcome. I don't think, and another thing I think we should really think as EMA is that why are we always 1 year behind FDA? We should think about this. Well, what are our procedures? We should work probably on speeding up in some ways. The procedures, I don't know if it's all on waiting for phase 3 data, but also procedimental procedures that make this process too slow. And speed, I think it's relevant also for patients, particularly with really relevant drugs. I think it's a different case. I don't know if you have with Pembro and a 15 or a 20% response rate in a certain scenario where it's more difficult to address. I think in that context, in a setting where you can do a controlled trial, you should do a controlled trial. And just my last comment is that if we are looking for biomarkers, I think controlled trials are important, particularly to be able to distinguish predictive from prognostic biomarkers and to be able to address cutoff issues and these sort of things. So I think controlled trials also provide some very relevant information in this regard.
Yes, this is Ramsey Dagger from Pfizer.
And a couple of comments. One I was going to suggest, but I'm glad the last two or three colleagues, individuals who spoke are touching on this, is that we focus on the bias, quote unquote, the bias that we think is inherent in single arm. But we sometimes forget about the potential bias against single arm. And one of the things I hope we discuss more later today, and more of it, I think it's already started from some of the comments, is that there are two related elements, right, in regulatory decision making. One is when you're guiding industry during development, we recognize that it's very important to have these conversations very early, so that at least if there's the possibility for the use of the single-arm studies as part of a development program, at least, regardless of whether for any particular indication it's going to be the only evidence or the main evidence, or whether it's part of a broader program that will at some point include randomized studies either for the same population or a different one. It's important to look at two elements, I think. One is there going to be a feasibility of using single-arm data for some registration purposes during the plan? And then, related to it, what would we be willing to consider from all that data in describing the labeling? And so, for example, when there's comments on the scales that can be used to describe clinical benefit, I'm not saying it's only the MEA, but around the world sometimes there's a concern that if we go beyond describing the traditional radiologic criteria, that somehow that introduces more subjectivity. And so even in the situations where there's a willingness to use single-arm data for at least for initial authorizations, we sometimes also see that the focus of the description of the benefit is that radiologically driven kind of information, when from the comments we're starting to hear, a lot of that benefit, that granularity of the patient's perspective of the benefit, partly comes from the tumor shrinkage, but there's a treasure trove of information, even in the single-arm setting potentially, about the benefit beyond simply the radiologic. So I just hope that the conversation continues not just about when would be the right circumstances, criteria for using single-arm data for some regulatory purposes, but also how much information could we describe? For example, even we talk about sometimes that, okay, the response criteria are fine for single-arm, but maybe describing time to event endpoint like PFS, maybe there's more concern about that. But for example, as Mace showed in our Crizotinib data, when you have a very big effect, maybe you can be more comfortable. And also, already, even in the standard criteria that sometimes we use, we talk about the duration of response. Well, duration of response is, in a way, has a time component to it. And yet we're very comfortable. It's important that we actually use that dimension to give more certainty about the benefit. So the theme is, I hope that all these comments around the patient perspective on the benefit, the other elements, that we don't forget about that and not just focus on should we use those studies or not, which are also important.